Hello there, this is Savannah here with the concept linkage. So we already know that in each and every plant there are thousands of genes that are present, and we also know that the number of genes that are present is more as we compare it to the number of chromosomes that is present in a cell, which means every chromosome possesses more than one gene in them. That is how the number of genes present is more than the number of chromosomes. But it was in the year 1903 William Sutton had expressed the probability that every chromosome must have more than one unit factor. Now we already know that the different characters are governed by the genes that are present in the chromosome. So the genes for different characters may be either situated on the same chromosome or it can be situated on completely different chromosome. So when the genes are situated in different chromosome, the characters that they could express in the upcoming generation can be together or they can be apart. So this depends on the chance alone, which means we know the Mendel's law of independent assortment, according to which the genes that are situated on the same chromosome or the different chromosome, they tend to segregate separately or assort separately. But what if the genes are situated on the same chromosome, or just say they are fairly close to each other or they are present close to each other? What happens? They tend to inherit into the next generation together. So this type of coexistence of two or more genes on the same chromosome is called as linkage. So in the year 1910, T.H. Morgan and his colleagues, they determined that genes are not completely independent as Mendel had thought when he carried out his experiments, but they tend to inherit in groups as Morgan and colleagues were carried out their experiments. So they say the genes they are linked to each other in a linkage groups. They are called linkage groups. So they tend to inherit in groups there. And then when these linked genes are acting in a particular inheritance, the phenotypic ratio, the F2 phenotypic ratio will not remain the same. That is the 9:3:1 will not be the same as Mendel had given us. So it will be different. It will be like altered. So they observe that in the same chromosome they are often transmitted together as a group, but this was not always so, and the crossing over between the chromosomes could occur to disrupt these linkage groups, which means there were linkage groups, which means the genes were linked in a particular chromosome, but they were not always inherited together. There were some phenomenon that is happening called crossing overs, which mean the crossing over of or the physical exchange of genetic materials could happen between the adjacent chromosomes, which would interrupt the inheritance of linkage gene. So the genes that are present on the same chromosome and that tend to be inherited or transmitted to the gametes together are termed as linked genes, because the DNA sequence that contain the genes is passed along as a unit during meiosis. It is not like genes will be separated and it will be gone to the, or it will be distributed to the gametes. It's not like that. When meiosis is happening, the DNA sequence will will get transferred as a unit, as a whole. So the genes, if they are placed closer or they remain closer in particular chromosome, there is higher probability that they will be inherited as a unit, since crossing over cannot happen between these two genes if they are closely placed. But if they are these two linked genes are not too close to each other, they are far from each other, then the crossing over was very common and they were not tend to be linked together. So the gene that is present on the same chromosome would not assert independently in such type of inheritance, which means where crossing over used to happen commonly. So such type of genes where they are linked together in a particular chromosome and they tend to inherit together are called as linked genes. And the phenomenon of these linked genes inheriting from a parental generation to the offspring generation is called as linkage. It's not only this Morgan and other people they gave the theory, not this particular definition. Morgan also defined linkage as the pair of genes of homozygous parents in the sense they have the same, similar or same type of chromosomal pairs, gene pairs. So here the pair of genes of homozygous parents tend to enter into the same gametes and to remain together, whereas same genes from heterozygous parents tend to enter into the different gametes and remain apart from each other. So he also further stated that the tendency of linked genes remaining together in original combination is due to their location on same chromosome. Alright, if they are very close to each other, they tend to be inherited together to the offspring generation, or if the location of these two linked genes are far from each other, they tend to inherit separately into two different gametes there. So according to him, the degree or strength of linkage purely depends on the distance between the linked genes in a chromosome. Now, Morgan, along with his colleagues, Castell formulated the chromosomal theory of linkage, uh, which is described as follows. So the genes which show the phenomenon of linkage are situated in the same chromosome and separated during the process of inheritance, which means whichever the linked genes or the linkage, the process is carried out by the linked genes that are present on same chromosome, and the linked genes that are present on different chromosomes were not inherited together. They were separated during the process of gamete formation. And the thing is, the distance between the linked genes determine the strength of linkage. If they are closely located genes, they show a strong linkage between them, and they tend to inherit together in offspring generation. And if they are located far away from each other, then there is weak linkage between them, so that the crossing over can happen between them, and then there is no chance of these two linked genes inheriting together into the offspring generation. And the genes are arranged in linear fashion. So these linked genes are always arranged in linear fashion. Only then they can inherit together. If they are present in other fashions, like they are present on different chromosomes, then there is no linkage as such. So there are different types of chromosome based on different criteria. So it is there based on the crossing over type, and it can be divided based on the genes that are involved, and it can be determined using the chromosomes that are involved. So there are different types of linkage. So based on the crossing over, there is this three type of linkage. One is complete linkage, incomplete linkage, and the third one is absent linkage or non-detectable linkage. Now, the first type is here during the synopsis of a cell division phase meiosis, there is exchange of segments that is taking place. Alright, in this condition, the possibility of separation of two genes that are situated close to each other is very much reduced. Alright, so these can't be separated so easily because they are placed very close to each other. So when these genes are closely associated and they tend to transmit together, this particular linkage is called as complete linkage, so that there is no crossing over happening between them because they are very close to each other and they completely inherit those two linked genes completely inherit into a single individual. So this type of linkage is called complete linkage. And the second type of linkage is incomplete linkage. So here, when linked genes are situated at long distance or at wider distance, so these two have space in between them, so these have chances of separation as crossing over can happen between these two linked genes. So as a result, what happens, they are not completely inherited, they are incompletely partially inherited into the gamete or the offspring generation. So this particular linkage where they are partially inherited is called as incomplete linkage. So in incomplete linkage, there is a chance of crossing over that is being happened. Now there is third type, which is called absent or non-detectable linkage. This is because here the two linked genes are placed at a very far distance, or the space or the physical distance between these two linked genes are very much far than compared to the incomplete linkage. So what happens, there can be there is a higher chance of crossing over that is being happened, and it tend to not inherit these two linked genes together. Right, so this means this cannot be detected as linked genes because it's purely a chance basis. These can get into two different gametes or two different individuals. So this is called absent or non-detectable linkage. And based on the genes involved, there are two types of uh, linkages, that is coupling phase and repulsion phase. So here this purely depends on whether all dominant or some dominant and some recessive alleles are linked together. So here in coupling phase, dominant alleles are present on same chromosome and their recessive alleles are present on the same chromosome. All right, so this is called coupling phase. And in repulsion phase, it is just a vice versa of the coupling phase. Here one dominant allele of some genes are linked with recessive allele of other genes. Okay, so these two are the homologous pairs of chromosomes. So here one dominant allele of a gene and here a recessive allele of another gene are present or linked together. So these are called repulsion phase. And based on the chromosomes that are involved, there are two types of linkages. One is autosomal linkage and the other one is allosomal linkage or sex linkage. So in autosomal linkage, this refers to the linkage of those genes which are located in autosomes, that is the somatic chromosome. If the linkage is observed in the if the linked genes are present in the somatic chromosome, it is called autosomal linkage. And then if the linkage genes are present or located on the sex chromosome, it is called allosomal linkage. So in sex chromosome, it can be either present on X chromosome or Y chromosome. And there is this concept which I mentioned earlier called linkage groups. So here the group of linked genes that are located on same chromosome is called linked group or linkage group. So here the number of linkage group are limited to each individual. All right, so the each individual have a limited number of linkage groups in them. So here the maximum number of linkage group that can be possible will be equal to the haploid chromosome number of an organism. Say for example, in corn the diploid chromosomal number is 20, then the linkage group that is possible is half of this diploid condition, which means the haploid number of chromosome will be the number of linkage groups that is possible in that particular individual. So here 10 linkage groups are possible in corn who has the diploid chromosome number 20, and 7 linkage groups in garden pea whose diploid chromosome number is 14, and same with the barley. 7 linkage groups are possible, that is the maximum 7 linkage groups are possible which have that has diploid chromosome number 14. Now, so linkage in maize, this is a good example of linkage. So this follows incomplete linkage here. So Hutchinson crossed a variety of maize having colored and full seed. So colored is this was governed by the dominant gene C, and full seed, this phenotype was governed by the dominant allele or dominant gene S. So this was crossed between this was crossed with a variety having colorless and shrunken seeds. So colorless was a condition that was governed by the recessive recessive allele C, and shrunken seed was a phenotype that was governed by the recessive allele S. So here when these two phenotype or these two parents were or these two varieties were crossed, then there was F1 plants that produced colored and full seed because the gene C for color is dominant over its colorless allele C, and the gene S, which governs the full seed phenotype, is dominant over the shrunken allele S. So in F1 generation there were both the allele that is being present in heterozygous condition. So what happens, all the F1 plants produced will be having the dominant phenotype, that is a colored and full seed. And to determine whether the genes are linked to each other, there were test cross that was being carried. As can be seen here, this is the dominant parent and this is the recessive parent, and this is the F1 hybrid here. So when this was crossed back with the recessive parent, that is the double recessive parent, which is shown here, this is the back cross here, there was gametes that is being produced. So here 4 types of gametes was produced because this was double heterozygous. So there were 4 different types of gametes that was being produced, and in double recessive parent there is single gamete that is being produced. And when these were crossed, the total number of 8,368 progenies were produced. So of which colored full progenies was 4,032 of 8,368 progenies, and colorless shrunken seeds were 4,035. So these are the number of progenies of the total progenies produced, and colored shrunken is 149 and colorless full seeds were 152 out of 8,368 progenies. So when this result was stated, and this is so clear that almost 96 to 97% of the progenies in the test cross was of parental character or parental combinations. Colored full was a dominant parent and colorless shrunken was a recessive parent. So 96 to 97% of progenies were of parental combinations, and only 3.6% of the combination that was obtained is new combination, which means only within these 3.6% of new combination crossing over have been occurred. That is why there is new combination that has been occurred here. So this clearly shows that the parental characters are linked together, and so because they are linked together at the time of gamete formation, uh, they are together assorted into a single gamete and they are carried to a individual there. So that is how each individual here has maximum of parental combination, and there is only few percentage of new combination that is being observed. So this shows the incomplete linkage between the genes that governs the colored and full seed and colorless and shrunken seed in maize. So there's another concept called arrangement of genes. So this is basically like there are 2 types of arrangement of genes, that is cis arrangement of genes and trans arrangement of genes. So this actually says where the alleles are present in the homologous pair of chromosomes. So in cis arrangement of genes, if the dominant alleles, that is A and B, of 2 linked genes are present on same chromosome and their recessive alleles, that is the alleles of these A and B, are present on the homologous chromosome, the arrangement is called cis arrangement. As can be seen here, these two are the homologous chromosomes. So A and B are 2 different linked genes. So here the dominant alleles are present in single chromosome and the recessive uh, genes are present, or recessive alleles are present in its homologous chromosome. This type of arrangement is called cis arrangement of genes, and whereas in trans arrangement it is just the opposite. It's like the transferring is happening here. So here in 2 homologous chromosomes, one chromosome have a dominant gene and other recessive gene. The same with the other homologous pair also. There is one recessive and the other dominant allele present in them. So this particular arrangement is called as trans arrangement of genes.